Hello everyone and welcome to this video session of chapter 2 part 2, and today we are going to learn about meiosis. What happens in meiosis? And as we learned in previous chapter how cell division gives two cells, today we are going to learn that in meiosis it gives 2 plus 2, 4 number of cells. And in order to learn about that, we need to know that meiosis, which comes from a Greek word which means lessening, which means that in mitosis we get 2 cells, but in here we are getting 4 cells, but the chromosome number is going to reduce, and that we will learn at the end of this video. It is a special type of cell division in sexually reproducing organisms which is used to produce gametes such as sperms, egg cells, and it involves 2 rounds of division that ultimately result in 4 cells with only 1 copy of each haploid. Additionally, prior to the division, genetic material from the parent and maternal copies of genetic material of each chromosome is crossed over. Crossing over means that they are exchanged and they create a new combinations of code on each chromosome, and later on during fertilization the haploid cells produced by meiosis from a male and female will fuse to create a cell with 2 copies of each chromosomes again, the zygote. So, what is meiosis? Meiosis is a process where a single cell divides twice to produce 4 cells containing half the original amount of genetic information. These cells are our sex cells: sperm in males, eggs in females. And during meiosis we have to take an account for 3 basic things, like that one cell divides twice to form 4 daughter cells, and secondly, these 4 daughter cells have only half the number of chromosomes of the parent cell. They are haploid. And thirdly, meiosis produces our sex cells or gametes, eggs in females and sperm in males. Meiosis can be divided into 9 stages because it has 2 types of meiosis taking place: meiosis 1 and meiosis 2. In meiosis 1, there is a reduction of chromosome number to half the number of chromosomes, and in meiosis 2 there is equational division like mitosis. They are divided between first time the cell divides in meiosis 1 and again they divide second time in meiosis 2. Now let us see what goes on in these various stages. So as meiosis 1 and meiosis 2, we can coincide this with mitosis. Mitosis has 4 stages: prophase, metaphase, anaphase and telophase. Same in this also we are going to learn about that, but it will, when you write it down, it will come as meiosis 1 and prophase 1. So in meiosis 1, what happens in interphase, which is the non-dividing phase, that the DNA in the cell is copied, resulting in 2 identical full sets of chromosomes. Outside of the nucleus are 2 centrosomes, each containing a pair of centrioles, and these structures are critical for the process of cell division. And during interphase, microtubules extend from these centrosomes. And now prophase 1. In prophase 1, the copied chromosome condense into X-shaped structures that can be easily seen under a microscope. And each chromosome is composed of 2 sister chromatids containing identical genetic information. The chromosomes pair up so that both the copies of chromosomes are together, both the copies of chromosomes 1 and 2 are together, and so on. The pair of chromosomes may then exchange bits of DNA in a process called recombination or crossing over. And in crossing over, a part of the chromatid is then exchanged with other chromatid in order for crossing over to happen. At the end of prophase 1, the membrane around the nucleus in the cell dissolves away, releasing the chromosomes. The meiotic spindle, consisting of microtubules and other proteins, extend across the cell between the centrioles. Now comes metaphase 1. In metaphase 1, the chromosome pairs line up next to each other along the center, which is the equator of the cell, as it also happens in metaphase of mitosis. So the centrioles are now at the opposite poles of the cell, with the meiotic spindle extending from them. The meiotic spindle fibers attach to one chromosome of each pair. So here the metaphase ends. Now comes the anaphase 1. And in anaphase 1, the pair of chromosomes are then pulled apart by the meiotic spindle, which pulls one chromosome to one pole of the cell and the other chromosomes to the opposite pole. But in meiosis 1, the sister chromatids stay together. This is different to what happens in mitosis and meiosis 2. In mitosis and meiosis 2, they are splitted apart and are then moved towards the opposite poles. But here they stay together. Now comes telophase 1. In telophase 1, the chromosomes complete their move to the opposite pole of the cell. At each pole of the cell, a full set of chromosomes gather together. A membrane then forms around each set of chromosomes to create a new nuclei. The single cell then pinches in the middle to form 2 separate daughter cells, each containing a full set of chromosomes within the nucleus. And then they are separated, as we learned in the previous video of mitosis, how a furrow develops and the cell, which is bigger inside, then forms a cleavage and then they are separated. And this process is known as cytokinesis. So now this meiosis 1 is over, and we end this. We understood that now the chromosome number has halved to n. In mitosis, each cell is termed as 2n number of chromosomes, but in this it is just a single n. That means it has the half number of 6 chromosomes in it. Now we have 2 cells, but how are we going to get 4? Because we know that in meiosis, this division gives out 4 cells. So now meiosis 2 comes. In meiosis 2, again these 2 cells will go on dividing and completing their cycle like meiosis 1, but it will be known as equational cell division, because here the cells are not going to divide for half number of of chromosomes, but then they are going to divide, as in like it happens in mitosis. So again the cell will go on from prophase to then metaphase 2, then anaphase 2, then finally telophase 2 and cytokinesis. And again this cytokinesis, when it happens, there will be 4 number of cells, but they will have half number of chromosomes. So once cytokinesis is complete, there are 4 granddaughter cells, each with half a set of chromosomes, which all are haploid. And in males, these 4 cells are all sperm cells. But there's a different thing about females. In females, one of the cell is an egg cell, while the other 3 are polar bodies, small cells that do not develop into eggs. So these polar bodies will not take part to become the full matured ovum or the female egg, but then they will be termed as polar bodies and they will have no longer part in reproduction. So till now we have understood and cleared the basic concept of how mitosis and meiosis happens, and now we are going to see that what is the significance of meiosis. We know that if we revise that meiosis is a process in which the parent cell divides twice into 4 daughter cells containing half the original amount of genetic information. That is, the daughter cells are haploid, which are termed as sex cells. So we know that there are features of meiosis, and we will go through them. Number 1 is it results in the formation of 4 daughter cells in each cycle. Then that all the daughter cells are identical to the mother cell in shape and size, but different in chromosome number. They will be similar in shape, similar in size, but the chromosome number will be half. And they are haploid, and the recombination segregation takes place in meiosis, and this process occurs only in the reproductive organs and results in the formation of gametes. And gametes are the male or female sex cells. So now let us look at the significance, that why meiosis is important. Meiosis is responsible for the formation of sex cells or gametes that are responsible for sexual reproduction. So this is the number 1 point. Number 2, that it activates the genetic information for the development of sex cells and deactivates the sporophytic information. Number 3, that it maintains the constant number of chromosomes by halving the same. That means it will divide it to half. And this is important because the chromosome number doubles after fertilization when male and a female gamete fuses together to form a zygote. And in this process, which is number 4th point, independent assortment of maternal and paternal chromosome take place. Thus, the chromosome and the traits controlled by them are reshuffled. So that is how the progeny that will be produced will have more different traits than their siblings. Number 5, the genetic mutation occurs due to irregularities in cell division by meiosis. The mutations that are beneficial are carried on by natural selection, but the mutations which are not important or which can degrade the whole life process will be taken out. So through natural selection, it is the pure breed that comes into place. And lastly, crossing over produces a new combination of traits and variations. I hope that by this you might have understood all the basic concepts. Now the only thing that we need to understand this is that when we talk about meiosis and how it happens, there are few things that come in mind, like what is a chromosome, or what is a chromosome number, or what is an autosome, and what is the difference between a sex chromosome and autosome. So all these things will be covered in the next video, and I would like all of you to look for the notes and other links in the description below, and I hope that this will enhance our knowledge skills and will help us to score better and better marks in our ICSE biology examination. This is Gagan Singh, your teacher, and thank you. Have a nice day.